Good morning, everyone. My name is Jim Maltby from Cray, Inc. Thank you very much for attending today's webinar. Really appreciate your presence. Here's a quick outline of what I'll be talking about this morning. First, Cryo-Em Basics. What is it? And in particular, why is it so challenging for computers? Next, what are the bottlenecks? Two big ones: too much data and not enough compute. Then I will describe a reference architecture that we have developed at Cray to solve these problems. And finally, conclusions. Okay, now on to some basics of Cryo-Em. Cryo-Electron microscopy, or Cryo-Em, is a way of teasing out 3D structural information of biological molecules by imaging non-crystalline specimens of single large molecules. And as many people on this webinar will be aware, won the 2017 Nobel Prize in Chemistry. Here's a very nice picture that shows the increase in resolution that has been enjoyed in the last few years in cryo-electron microscopy. On the left-hand side, you can see a sort of a fuzzy blob-like structure, which was the resolution attainable just until about 5 years ago. However, advances in direct electron detectors, or DDDs, and mostly new software and new compute power has enabled the resolution on the right. It's a huge difference, huge increase. And as you can see, you can begin to get nearly to the level of resolution of individual atoms. Current limit seems to be about 2 angstroms. So before this resolution revolution, Cryo-Em was sort of referred to dismissively as blobology, but now it's becoming a very important tool. My title for this slide is Better Than Crystallography, and that's probably a bit cheeky, actually. In fact, it's much better than X-ray crystallography for certain types of biological molecules that do not crystallize at all. So, a very challenging procedure, but it works very well for those molecules that do not crystallize. Typically means that they're flexible and more complex, and larger molecular complexes of above about 150 kilodaltons. Okay, so why do we want these pictures at all? This is a very new field called structural biomechanics, and understanding the shapes of these molecules tells you what their function is. I'll show you a good example of that. This 3D structure is absolutely crucial. It shows where drugs can target, how these molecular machines work, and the more accurate, the more valuable that this information is.